Good evening. Sorry for the small glitch due to the internet. Today's topic is biological techniques, mainly DNA and protein based. So I will cover the DNA based, the PCR, quantitative PCR or real-time PCR, and the protein based, that is the ELISA and radioimmunoassay. So if we look, the DNA testing methods are based on the premise that each species carries a unique DNA sequence, and there are the unique sequences can be identified and used to differentiate the organisms. The DNA-based techniques, they provide a very robust approach for food authentication because DNA is relatively stable even after processing. You can make biscuits or cakes and yet you can isolate DNA from there. Since we have not had any webinar on DNA, let me just introduce DNA. So where do you find? Okay, shall I start?
Yeah.
Okay. Sorry for the glitch. Let's start again. The topic for today is biological techniques, the DNA and the protein-based methods, which mainly will involve PCR, the real-time PCR or qPCR, ELISA, and radioimmunoassay. So, So the DNA testing methods are based on the premise that every species carries unique DNA sequences, and using these unique DNA sequences, we can differentiate one organism from the other. The DNA-based testing are very robust approaches and very useful, not only in forensic science, because mainly it is used in forensic science, but it is also very useful for authentication of food. And the reason being that even at very high temperatures, DNA is relatively stable. So if you have processed the food, like baking biscuits or cake, etc., you can still isolate DNA, that is the component DNA of the ingredients, and you can analyze them. So where is the DNA? So if you look at the DNA, you can get it from any part of the living organism. It could be the leaf, the root, and in the humans, it could be the skin, the blood, etc. So if you look at the organism from the leaf, what you see is a whole group of cells. And it is in the cells that you know that all the cells, all the leaf-setting cells have got a nucleus. And in the nucleus, you have the chromosome. And the chromosome has got the genes. And the genes are nothing but a set of DNA. And this DNA is nothing but strands of these four alphabets, A, T, C, G. That is the nucleotides or the base pairs that we call them. So we all know that DNA is the blueprint of life and that it determines the characteristics of all human organisms. Like, you know, a cat is a cat and a dog is a dog. So a cat cannot give birth to a dog and neither can a dog give birth to a cat. So it determines the characteristic of every living organism. The DNA structure, it is a macromolecule and it is composed of the four-letter nucleotides which are referred to, as I mentioned, A, T, C, and G. The order of these four nucleotides, A, T, C, and G, actually determine the characteristics of the organism. And if you look at the human body, there are greater than 3 billion of these nucleotides, which make up all the genes. So in the nucleic acid, let us look at the basic structure of nucleic acid. The nucleic acid is a polymer. It is a polymer of nucleotides, just like how a protein is a polymer of amino acids. What are these nucleotides? What are these nucleotides? So these nucleotides contain a nitrogen base. They contain a pentose sugar. The ribonucleic acid contains ribose, whereas the deoxyribonuclease contains deoxyribose, that is a pentose sugar, and it has a phosphate molecule. There is a direction in this because you have the phosphate molecule and you have the 5' and the 3' end, we will see. The DNA and the RNA together make up about 15% of the dry weight of cells. So what are these nitrogen bases? That is the alphabets that we talk about. So if you look at DNA, you will find the adenine and guanine, which are the purines. They have 2 rings. And then you have the pyrimidines, cytosine and thymine in DNA, and you have uracil in RNA. So how is the pairing taking place in that? Because you will see that in the DNA structure, A will pair with T and guanine will pair with cytosine. So how does this happen? So if you look on the right-hand side over here, it is mainly due to the hydrogen bonding between the bases, that is the bases of the nucleotides, there will be hydrogen bonding. So between adenine and thymine, you see there are 2 hydrogen bonds. And in the case of cytosine and guanine, you see there are 3 hydrogen bonds. So you see a hydrogen bond forms between the nitrogen and the hydrogen over here. So it is these bonds that keep the DNA, double-stranded DNA, together. So next, if you look at the single strand of the DNA, you see that how these bases are linked in the single-stranded DNA. So what you see is that every 3' hydroxyl group of the ribose molecule will be in a diester bond with the 5' ribose molecule of the next nucleotide. So what you have is a 3' 5'. So you see between these two, you have got a phosphodiester bond. That means if you have a phosphoric acid here and a OH here, a water molecule is removed and you have a 3' 5' phosphodiester bond. The nucleic acid has got direction because finally at one end you will see the free 3' end, that means a free OH, and at the other end you will see the free 5 hydroxyl group. So DNA has got direction and it is read from the 3' end to the 5' end. So a very simple representation of this shows the sugar backbone, and it is the bases which point out, and it is these bases of the 2 strands which will interact with each other with the hydrogen bonds that we just looked at. So this is how the DNA gets its double-stranded nature. So a nucleic acid polymer, it's got a free 5' phosphate group at one end and a free 3 hydroxyl group at the other end. And it is read from one end to the other. So we read this as ACGT. DNA is double-stranded, as I said, because of the complementary base pairing, the base pairing of A with T and C with G. And the double-stranded structure, you know, was discovered by Watson and Crick, that is, and it is known as the DNA helix. And they have shown that the DNA turns, there is at every 3.4 nanometers, there is a turn, and the distance between two bases is 0.34 nanometers, meaning that every turn has got about 10 bases. The purine-purine pair, it cannot pair. That means adenosine cannot pair with guanine because the distance is too far apart. And pyrimidine, that means the cytosine and thymine cannot pair because it is too narrow. And it is only one purine and pyrimidine pair which is just right. And that is adenine with thymine and cytosine with guanine. So what is DNA-based testing? DNA-based testing actually came out, was born out of an understanding of the chemistry of DNA replication. You know that in normal circumstances and in natural circumstances, in all plants and animals, DNA replication takes place, where the double-stranded DNA will become two single strands. The single stranded DNA template and a free 3-hydroxyl group at the other end will incorporate the nucleotides and form a new DNA strand. And an enzyme, DNA polymerase, does this job in the living beings. So the polymerase chain reaction actually mimics the DNA replication. That is, if you look at this as the DNA double helix, in the first denaturation step, the hydrogen bonds break, and then each strand of DNA acts as a template for a new strand. And so what you get is from a single strand of DNA, you get two strands of DNA or two daughter strands which are exactly the same. So based on replication, the polymerase chain reaction, it mimics the DNA replication. But in the case of DNA polymerase, it is found that the DNA polymerase, which is found in in most of the living creatures, is not stable to heat. And so therefore, the PCR reaction or the replication could not be mimicked outside. However, the reaction was revolutionized by the discovery of an enzyme Taq DNA polymerase, which is stable at very high temperatures and could be used to mimic DNA replication. So what is DNA-based testing? It is the selection of a unique DNA sequence or a sequence between two organisms which differ, designing primers for that unique sequence, then isolating DNA from the food or from the organism or whatever is the sample for analysis, duplicating that DNA because the DNA isolated is in such minute quantities that you cannot detect it, duplicating that target sequence based on replication, and in analytical terms, we call that amplification. So it is amplified by changing the temperature. The enzyme that is used to build the DNA molecule is known as the Taq polymerase. It is thermostable, and the instrument that we use is a thermocycler, but everyone calls it a PCR machine. And then the results that you get can be either quantitative or it could be qualitative. So in a flowchart for the polymerase chain reaction or PCR that is there, what you need is the test material, and it is very essential that you have the standards, two standards, that is a positive control, you are sure that the DNA is there, and a negative control, you know that that DNA is absent. So the first step is DNA extraction, and I will tell you how we do that. Then it is checking the purity of the DNA, checking whether there are inhibitors of the polymerase chain reaction there, then doing the PCR amplification in a thermocycler, and then doing either DNA electrophoresis on a gel or using the real-time PCR to amplify the DNA into amounts that we can visualize, and then interpretation and documentation. So it is a long protocol, starting with the extraction of DNA, purity, and that. Before that, we need to identify the DNA sequence that you want to amplify. That is the unique sequence that you want to use. So what is needed for PCR is the template. So the template is the DNA, and you will be isolating the DNA. The next is primers. That means 2 short specific DNA sequences which will match the left and the right end of that unique target sequence that you want to amplify. And these are called as the forward primer and the reverse primer. And we need nucleotides, which are the building blocks, just like the bricks of a house. So we have DATP, DCTP, DGTP, and DDTP. We need magnesium chloride as an enzyme cofactor. We need a buffer to maintain the pH. And of course, we need mainly the DNA, the Taq DNA polymerase, which is a thermophilic DNA polymerase which has been isolated from an organism, Thermophilus aquatis. And so that is why it has got the name TAQ. So let me give you an example of how to identify a sequence. So this, if you want to identify or you want to check the adulteration of basmati rice, if you look at the metabolic metabolic cycles and the pathways that occur in the basmati rice, if you look at the cycle that is responsible for the aroma of basmati rice, there is a gene which is called the butyraldehyde dehydrogenase, which we call as the BADH2. And it is this gene which is responsible for the aroma of basmati rice. So if you compare the sequences of the BADH2 gene from the non-fragrant rice and the basmati rice, we can see there is a difference where there is a deletion of about 8 base pairs between the two, and there are 3 changes. So this sequence of the fragrant rice is unique, and it differs from the non-fragrant rice. So therefore, we can try to amplify this sequence. So if we design primers to amplify this sequence, we will see it only in basmati rice, and we will not see it in the non-basmati rice. So in the case of DNA-based methods, you have to identify the gene or the sequence which will differentiate A from B and B from C. For example, if you're trying to look at horse meat in the burger, you have to look at sequences which differ in horse and beef and use that gene or that bit of sequence to identify the horse gene. So you design the primers. We generally design a forward primer and we design a reverse primer based on the sequence that we want to identify. So generally the primers are very short sequences of about 16 to 18 base pairs, which will actually match directly. There is a direct base pairing with the original DNA. So then coming to so now you have identified the sequence, you have made the primers. So the next step is to extract the DNA. So in DNA extraction methods, you know that the DNA is in the cell and that too in the nucleus. We either isolate mitochondrial DNA or we isolate the genomic DNA. So generally in the technique, there are several techniques and several kits which are available. The first step is to lyse the cell, that is to break the cell wall so that the contents of the cell wall are spill out. Then with the lysed cells, because you want to isolate the DNA, which is a nucleotide, in the cells, you know, we have a lot of protein, we have a lot of carbohydrate, and we have a large amount of fat. These are the macromolecules, and we have some RNA. So we need to get rid of all these macromolecules. So you do a digestion with a protease to break down the proteins into amino acids. You do a digestion with RNAs to break down the RNA. You do a digestion with alpha amylase to break down the carbohydrate. Now, in the cell lysate, you have broken down all the macromolecules and only DNA is left intact. The next step is to precipitate the DNA. We can precipitate the DNA using chloroform, phenol chloroform. We precipitate the DNA. You can wash the DNA, and once you have extracted the DNA, we need to evaluate the purity of the DNA, because the success of a PCR based reaction, whether it is endpoint PCR or real-time PCR, depends on the purity of the DNA. So we can evaluate the purity of DNA by agarose gel electrophoresis, or we can evaluate the purity of the DNA by the ratio of the absorbance at 260 by 280. This ratio should be 1.8. If it is more than 1.8, nearing 2, you know that you have RNA contamination. If it is less than 1.8, if it is about 1.6 or so, you know that you have protein contamination, because this A280 represents the protein and this absorbance at 260 represents the RNA in the solution. So this is an overview of DNA extraction. So we grind the material, whether it is food, plant, root, etc., to a fine particle size. Generally, you can do this with liquid nitrogen. You can grind it to a very fine particle size. You suspend it in buffer. You add the enzymes proteinase K, alpha amylase, RNase, and incubate it for about an hour to digest all these macromolecules. Then you precipitate the DNA using isopropanol or phenol chloroform, etc. You centrifuge, so your precipitate will be DNA. You wash the DNA pellet, you dry it, and you dissolve it in some buffer, which we generally call the TE buffer. So this is the general protocol, and it varies from tissue to tissue. If you're using plant tissue or things, sometimes you might need to use harsher conditions to break the cell wall. So once you have the DNA, you analyze the DNA for its purity. You can do this with agarose gel electrophoresis. So what you need to get is a compact band, that because the DNA is very large, you get a compact band. And if there is any shearing because of the procedures that you have used, you will see a lot of smearing. It is stained with ethidium bromide, and the DNA, if you have an appearance like this, the DNA is pure. So what you see over here is wheat DNA, this is pDNA, human, fruit fly, and yeast. Now, determining the DNA concentration, as I said, you use the formula A260 by A28. The purity by using A260 by A280. But if you want to determine the concentration of the DNA, you take the reading at 260 nanometers, you multiply by the dilution factor, and one absorbance unit, that is one optical density or one absorbance unit at 260 nanometers, is equal to 50 micrograms of DNA, and one OD is equal to 40 micrograms of RNA. So depending on the absorbance, you can calculate the concentration of DNA that you have. Determining the purity, as I said, the ratio for A260 by A280, ideally it should be 1.8. If it is less than 1.6, you have protein contamination, and if it is greater than 2, you have contamination with RA. So this absorbance at 260 by 280 is a measure of the purity of the DNA. So the polymerase chain reaction, let us look at that. It actually is a license to do molecular biology, because without the enzyme polymerase, you cannot do any molecular biology. Without PCR, any molecular biology experiment is redundant. So a key central technique that has revolutionized molecular and consequently cell biology. So the person who has credit for this is Kary Mullis, who discovered the polymerase chain reaction, and it was possible because of the discovery of this enzyme, that is the Taq polymerase from Thermus aquaticus, from the hot springs. So it is abbreviated T for Thermus and AQ for Aquatis. It is very heat stable, so it is stable even at 94 degrees centigrade, and the optimal temperature for its reaction is 72. So these temperatures are very important. Please remember these temperatures when we see how the PCR cycles or reaction is done. So how does the polymerase chain reaction take place? So in the polymerase chain reaction, you first have the double-stranded DNA. To that, you have added the DNTPs, that is the DATP, DCTP, DGTP. You have added Taq polymerase, and you have added the forward and reverse primer, and we have magnesium, which is the cofactor required for this reaction. So in the first step, now please watch, this is the DNA strand. So in the first step, you have denaturation. So the strands will move apart, or they will denature. In the second step, the primers will anneal. So you see the two primers. So the primers are annealing. And I said the primers have to be exactly complementary to the strand. So you see the complementarity. So only when there is complementarity between the two, the DNA will bind. So this reaction is almost like building a house. And here the DNA, the double-stranded DNA, is like the foundation of the house. And the primers that you are adding are the stones that you have put to keep the foundation intact. The DNA polymerase is like the mason, and the ATGC is like the bricks that you add to build the wall. So the first thing is that in the foundation, the annealing of the primers is very important. So you have to design the primer so that there is complete complementarity here and not in any other region. So the next step is the enzyme recognition by the enzyme polymerase. So the polymerase enzyme comes and it recognizes these Y-fork-like areas. So the DNA polymerase recognizes these areas, then it needs to react and for reaction it requires the magnesium cofactor. So the magnesium cofactor comes, and once the magnesium cofactor comes, the building of the DNA strand with the individual bricks or the individual DNT pairs takes place very fast. So that step is called extension. So the denaturation step we do at about 94 degrees Celsius, and the extension step we do at 72 degrees Celsius. I asked you to remember what we had seen in the previous one, that it is thermally stable even at 92 degrees Celsius. So when you heat the PCR cycle to 94, the enzyme does not get denatured. And when we reduce the temperature to 72, you see the reaction will take place, or extension takes place. So what happens in extension? You see that these bases will be complementarily added, and two new strands of DNA will be formed. So from a single stranded DNA, what you get is, from one copy of a double-stranded DNA, you get two copies of the double-stranded DNA. So now the strand is duplicated. So this is the basis of the PCR reaction. And we can continue this. So now you can do next a denaturation. These two strands will separate. These two strands will separate. The reverse and the forward primer will anneal. Then you will have polymerase recognition, and then you will have extension. And these two DNA will now become 4 DNA. So you have one copy of a DNA, then you have two copies of a DNA, and then you have 4 copies of a DNA, and then you will have 8 copies of the DNA. So these cycles are repeated. So first the denaturation for the DNA strands to separate, then an annealing step for the primers to anneal, then an extension step, that is at the optimum pH of the Taq polymerase at 72 for the strand to complete, and then again the next denaturation step takes place. So now the first denaturation, and now this cycle can keep continuing, and from at the end of about 25 to 35 cycles, you will see that the amount of DNA increases by 2 to the power of 25. There is an exponential amplification of the gene. So theoretically, what happens is, in the first cycle, you get 2 copies, second cycle, 4 copies, in the third cycle, 8 copies, in the fourth cycle, 16 copies. And if you look after 30 cycles, we have this large number of copies. So we have amplified the DNA several fold, and this DNA can now be detected or visualized. So if you compare the PCR reaction with that of photocopying, because it is almost like that of a photocopying machine, you will see the only difference is the PCR machine, the photocopying machine can make one copy at a time, whereas the DNA, the Taq polymerase and thing, makes 2 copies. There is an exponential, and so you have millions of copies of the DNA which can be detected. So this is the actual illustration of the wanted gene, that is the template DNA, and you have the primers. So now you see it tells you how you get that. So at the end of 35 cycles, you will have 34 billion copies of the DNA. So an example of using this PCR reaction is for the detection of GM, that is the GMOs. So here in a GMO, the difference between a non-GMO and a GMO is only this new DNA sequence which has been inserted into the GMO. So you can target any one of these regions from the promoter region to the TNOS terminator region and try and amplify this. So what you will see is you will get amplification only in the GM plant or the GM food, and you will not get amplification in the non-GM food. So if you look at this gel, so first one is the premise control to say that you have not got it from the water. Now, number two is a control biscuit, which does not contain the GM soya. So you see there is no amplification of this gene region from here to here. There is no amplification of this region. However, if you look at number 4, which is a biscuit we have labeled GM1, and another biscuit which is GM2, and the last lane is the positive control or GM soya. But when using DNA-based testing, these controls are very important so that you do not have any false positives or false negatives. So you need to have a positive which contains that DNA sequence and a negative which does not contain that DNA sequence. So here are two gels, the endpoint PCR, one which shows you the amplification region from here to here, and the other is to show a region from here to here is amplified. But in all cases, you see, we have the control biscuit where GM soya is not added, and you have the positive control, which is in lane number 4, the positive control. And to find out the size of the DNA that you have amplified, you put what is known as the DNA ladder. And based on the ladder, you can tell the size. So this is 500 base pairs. So this is approximately 210 base pairs for the amplification. But the disadvantages of just agarose gel, the PCR with the agarose gel electrophoresis is there is poor precision. Sometimes you might get amplification, a higher amplification, and you could also get primer-dimer formation. It has low sensitivity. It has a very short dynamic range. There is low resolution. It has to be done manually. It cannot be automated. It is based only on the size, as we have seen, and the results cannot be expressed in numbers. It can only be yes or no. Ethidium bromide is used for staining. It is carcinogenic, and also it is not very quantitative. And you get the same endpoint results with different initial amount of template. So if I start with 5 nanograms of DNA, or I use 40 nanograms of DNA, at the end of 35 cycles, almost all will look the same. So the amount or the quantification of the DNA is not possible of the amount of the parent DNA which is there in the sample is not possible with endpoint PCR. So to overcome this, the technique called real-time PCR has been discovered, and here the amplification of the target DNA sequence can be followed at every cycle by monitoring the product form. So if we go back here, so it will monitor in the first cycle and tell us there are only 2 copies. In the next cycle, there is thing, and you will be able to monitor in each cycle how much DNA is formed, unlike the endpoint PCR. And the detection strategies are based on fluorescence in real-time PCR, or also known as qPCR. So the most popular qPCR reaction is that of using a probe called Tachman. It employs a fluorogenic probe, which is in addition to the gene-specific primers, that is the forward and the reverse primer. And this fluorogenic probe has at one end a reporter dye and at the other end a quencher dye. The distance between the reporter dye and the quencher dye is such that the quencher dye will quench the fluorescence of the reporter, and initially you will see zero fluorescence. So let us see how this real-time PCR takes place. So the real-time PCR, the components of the real-time PCR are almost the same as that of NPCR. It needs the DNA, that is the template DNA. It needs the DNTPs. It needs the Taq polymerase. Okay, it needs buffer. It needs magnesium, and it needs the two primers, the forward primer and the reverse primer. In addition to these, unlike the other PCR, it requires a third primer, what we call as the probe or the fluorogenic probe, which has a reporter gene at a reporter dye at one end and a quencher dye at the other end. So how does real-time PCR take place? So initially you see this is your DNA template. You do a denaturation step, so the double-stranded DNA will separate. So there is denaturation. In the next step, you see you have an annealing step where the forward primer will anneal in this region and the reverse primer will anneal in the opposite region. In addition, the probe that you have made, which has a reporter gene at one end and a quencher gene at the other end, the distance of this probe is such that the fluorescence of this reporter is quenched, or quenched is taken away by this quencher. But this also anneals to almost somewhere in the middle of the DNA template. So due to the target specific sequence, this probe anneals specifically to the target DNA between the forward and the reverse primer. So you see, if this is the forward primer and the reverse primer, this target, the probe binds in the middle of that. Now, in the next step, when extension takes place and the double strand is being built, you will see that the Taq polymerase comes and the extension of the DNA strand takes place. At that time, the 5' exonuclease activity of the DNA Taq polymerase that we use will cleave this probe. So it will hydrolyze this probe, and when it hydrolyzes the probe, it can only do that when the extension has reached the region where it has primed. So once it comes here, the Taq polymerase cleaves this internal probe, and when it cleaves this internal probe, the reporter and the quencher will now move away. They will move far apart, and when they move far apart, the quencher can no longer quench the fluorescence of the reporter, and fluorescence will be the fluorescence which will be measurable. The fluorescence will be measured in the instrument. So the emitted fluorescence will be measured in the instrument. So the 5' exonuclease activity, it has cleaved the quencher from the thing, and now you see fluorescence is emitted, and that signal can be measured. So what are the dyes and quenchers that you use? Again, the dyes and the quenchers that you use, they have to have an excitation and emission wavelength which is very far from each other. Commonly, in most real-time PCRs, we use the common choices are FAM, Wick, Hex, and Joe as the reporter, and we use TAMRA and NFQ as the quencher. So if you look at this, FAM has an excitation wavelength of 492 and an emission of 516. And if you look at TAMRA, it has an excitation wavelength of 556 So because the excitation wavelengths are different, the fluorescence measured will be only that of FAM when you have the FAM and TAMRA probe. So it's very important when you're designing these experiments to see what you put as the reporter and what you put as the quencher dye. So in the real-time PCR, very similar to the PCR machine, you have the same PCR machine, but except you have a probe which at the end of the extension will measure the fluorescence. So if you look at the amount of DNA, the amount of DNA that is formed will be the same as that in endpoint PCR. So you have one cycle, two cycle, in the first cycle 2, second cycle 4, third cycle 8, fourth cycle 16. So this is exactly the same as you see in endpoint PCR. The only difference is that the fluorescence will be measured. So you see, as you go into the PCR cycle number, when you go, the fluorescence, that means the quencher and the reporter are still very close to each other, and it takes almost up to the 24th or the 25th cycle for the fluorescence to increase. So now you see that the amount of DNA, you can easily see the amount of DNA increasing after every cycle. However, towards the end of the PCR amplification, you see that there is not much change. So here you have 1400, it has become 1500, 150 or 4. So it has reached a saturation. So what you do at endpoint PCR is we measure the DNA here. So whether you have 1 nanogram DNA or 40 nanograms DNA, at the end we will have this, and so we are not differentiating. We cannot differentiate or quantitate the amount of DNA. So if you look at, if you plot the amount of DNA versus the PCR cycle number, you will see that there is an exponential release, and you will see that there is a tapering off of the thing, where this is the saturation. Okay, and then we set about, this is exactly what you see in the PCR machine, which gives you the cycle number versus the relative fluorescence intensity. So you see, even though DNA is formed, the fluorescence, because the probe is still bound, you do not see the fluorescence. But as the probe gets hydrolyzed, the fluorescence is visible. So this is used to quantify the amount of the DNA, that of the target DNA that we are interested in, or the analyte. So if you look at GMO analysis, so if you see, when you have less GMO, it takes much longer to amplify. When you have more GMO, that means more of the GM DNA, it amplifies much faster. So this is how you can quantify the amount of DNA or quantify the amount of the analyte. You can plot the percentage of the GMO versus the cycle number, and from that you can determine the unknown, as you will see over here. So here is the PCR, the cycle, the amplification plot of the real-time PCR. So what you have over here is, this is 0%, this is 0% DNA. This next one is 0.039% of the GM DNA. The blue is 0.023, the green is 0.18, the cyan is 1.6, and this is 9.9%. These are the standards. Just like how you make a calibration curve for your sugar analysis or your fat analysis, any analysis you make a calibration curve. In real-time PCR, also a calibration curve is made using known quantities of the analyte. And then you see this red line is the unknown sample. So you have to measure the analyte in the unknown sample. So in real-time PCR, we plot not the CT value or the cycle number versus the percent GMO, that is the log percent of the GMO. You get a straight line with a regression, and now you can use that to determine the unknown. So if you see the unknown, the amplification is at about, if you see it is at about 30, it's about 34 or 35, and if you see 34 or 35 corresponds to 0.72%. Also, if you look at the amplification plot, you see that it amplifies between 1.6 and between 0.18, which is almost the. So this is how you use real-time PCR to quantitate. It could be DNA, it could be, say, Salmonella, it could be Staphylococcus. So these real-time PCR or qPCR can be, the primers can be designed to amplify different genes in the microorganisms. You can isolate the DNA, enrich, isolate the DNA, and you will be able to tell how much of the microorganisms are there. It is not only used in GMO analysis, it is used largely in pathogen analysis to quantitate the pathogens in the food. And this is quite reliable, very reliable, because you are targeting a particular gene in the pathogen. So this is how an actual plot, this is a plot from the instrument. So you see here, we have a GMO specific gene and we compare it with its plant gene. Because in GMO analysis, we have to always give the reference gene or to show that the gene is coming from a particular plant. Like for soybean, we have to amplify the lectin gene to show that the DNA that we have amplified is that of soya and not that of jawar or corn, etc. So for maize, we will amplify the invertase gene. So we have got what is known as reference genes or housekeeping genes, and the GMO specific gene is compared with the housekeeping gene. So the PCR reaction is also used other than PCR and RT-PCR. Other types of DNA markers are also used to do speciation, to find out the authenticity, and to tell if there is adulteration. These include the mitochondrial DNA and looking for the gene cytochrome B, the tRNA or the rRNA. You can determine the species and thing by restriction fragment length for polymorphism, or by amplified fragment length for polymorphism, or RAPD, or variable number tandem repeat, or simple sequence repeats. The simple sequence repeats have been used to actually to differentiate the basmati rice, and the European Union uses the SSR to determine the authenticity of the basmati rice that is being exported from India to the UK and the EU. So what is RFLP marker? It is one of the oldest DNA marker technologies. It relies on a difference in DNA sequence between two samples that creates or removes a restriction site. So what is a restriction site? A restriction site is a site of 6 base pairs, which is very specific for the cutting of by the enzyme which are called as the endonucleases or the restriction enzymes. And these sequences that you see are palindromes. So this is a palindrome. So whether you read forward, so you see this is GAATTC, and if you read from this side, this is GAATTC. This type of a sequence which reads the same in both the directions. Examples in English are madam or Malayalam or onion. You have O-N-I-O-N. No, onion is not there. It's madam, and Malayalam also, you see, whether you read from left to right or from right to left, the spelling is the same. And that is called as a palindrome sequence. So in DNA, it is a palindrome sequence between the two strands. It's not in the same sequence. And these sites, the restriction enzymes have got very specific sites. So we can isolate the DNA, and you can actually cut it with the restriction enzymes and then look at the size of the fragments you get. So the other marker is the RAPDs. So here also, this is the random amplification of DNA. We amplify the DNA by using random primers, which are all of 10 base pairs, and we use only one primer per reaction. These primers will bind randomly to the DNA, and they get amplified, and you will see that you get a whole host of different products. Now you can compare these products because we have got databases by which you can compare these gel patterns, and you can tell to which species it belongs. Then PCR is also used for the DNA barcoding approach. The DNA barcoding approach is to give a barcode for every plant or every organism. So what we do is we select universal markers, and you extract the DNA. You amplify the DNA by doing a PCR, and then the target amplified regions, you sequence them by doing DNA sequence, and then you compare that sequence with the reference database, and comparing it with the reference database, you can identify the species. Nowadays, the databases for all whole genome sequences are available, so you can identify the species from the DNA sequence. So now coming to the protein-based detection. The protein-based detection depends on the protein, that is the antigen antibody reaction. And in these reactions, the antibody is usually tagged with a label. And when the protein binds to the antibody, the label, you will have a reaction, and that reaction will tell you whether it is positive or not. The lateral flow strip testing of using these protein-based methods is fast, cheap, and it is insensitive to thing. And it is a quick test called a test strip, and you can also quantify by using ELISA. So and in the protein-based assays, we have the technique which is known as enzyme-linked immunosorbent assay, or ELISA, and the term was coined way back in 1971. It is similar to radioimmunoassay, except there is no radio label over here. It can be used to detect antigen and an antibody. It is very sensitive at picogram levels. They use monoclonal antibodies, and the ELISA can be run both in a qualitative or in a quantitative format. So the premise behind ELISA-based testing is that you have an antibody against the analyte, so antibodies have to be made. Now, when the sample containing the analyte is introduced, the antigen binds to the antibody. The target antigen, you have a second antibody which is labeled with an enzyme, and that binds to the target protein. And once it binds to the target protein, you can determine or quantitate it by giving the substrate to the enzyme, because most of the antibodies that are used are labeled, or the proteins are labeled with an enzyme. An enzyme has been attached to them. So, what is an antibody? We have seen this when we talked about allergens. The antigen is a substance that is recognized as a foreign antibody by the immune system, and the immunoglobulin is produced in response to the antigen. So if you look at an antibody, you have the antibodies generally have this Y shape, which has a light chain and a heavy chain, and the end of the chains have got the antigen binding site, which are very specific for the antigen, just like a lock and key mechanism. So if you have an antibody, it's got the antigen binding site. If we put a whole lot of analytes or antigens, the only analyte to which this antibody is specific will bind. So it is very highly specific by like a lock and key system. So you see this blue antigen cannot bind, neither can this green antigen bind. Only this antigen can bind because it fits in exactly into the antigen binding site. And in fact, the antibody has been raised against this antigen. So in the lateral strip format, that is the dipstick test as we call it. I don't know how many of you have seen it, or if you have looked at the glucose strips to determine the urine in glucose, or if you have heard of the pregnancy test kits where within 5 minutes you get an answer yes or no. The lateral flow strips formats are available for a number of antigens, especially for the proteins of the genetically modified organisms, that is the herbicide tolerant soya against the EPSPS gene, and the insect resistant maize against the Cry1AB protein. So what is it that happens in a lateral flow strip? So you see, this is a lateral flow strip that you see. So if you look at a cross-sectional view of this lateral strip, at the bottom of the lateral strip, you have got the labeled specific antibodies which are immobilized at one end of it. Now, this antibody is specific for the Bt protein. So if I take a sample containing Bt protein and I put this strip into that, the interaction, because it is an antibody against Bt protein, the Bt protein will bind to the antibody. So the Bt protein which is bound to the antibody will move along the lateral flow strip along with the unlabeled, that means antibodies to which Bt has not bound. They will now move into a region, a second region, where there is a second specific antibody which again recognizes the Bt protein, and this is bound to an enzyme for a reaction, and if GMO is present, a reaction will occur and you will see a line here. The unbound antibodies which are there will move down the line and bind with where they are captured, and you will get a control line. So the lateral flow strip format is designed in such a way to tell you whether the strip is working or not. So if you have a positive, you get 2 lines. If you have a negative, you must get 1 line, because these labeled antibodies which are there will not have bound to any protein, and they have to move right across the lateral strip and only bind with the excess antibody captured here. So this is a fail-safe system which has been incorporated into the lateral flow strip, but it is based on an antigen-antibody reaction. So here are examples of the lateral flow strip. These are flow strips specifically for the CP4 EPSPS protein, and it is used to detect the herbicide-tolerant soya. So you can crush the seed, shake it vigorously, let it stand or centrifuge it, and just dip the lateral flow strip, because these lateral flow strips will also give you the direction of flow. So now what you see over here is that in all the strips you are seeing the test line and you are seeing the control line. So therefore, you are not seeing the test line in the 0%. However, you can faintly see it in 0.01% and you can see it very well in the others. And the control line is visible in all the 5, showing that the strips are working. So this is a 5 to 10 minute test and can be done in the farmer's field, and they can identify the GMO from the non-GMO seed. Here is another lateral flow strip test. This is for the insect-resistant maize. So these test strips are against the Cry1AB gene. And if you look at these strips, the antibodies you see, we can only detect 5%. You faintly see in the 10%. But what is more important to see over here is that you are not seeing the control line in this strip, showing that this strip is invalid. Even if you get a positive, you cannot use this to indicate that it is a GMO. Once again, in lateral strip flow tests, especially for GMO analysis, you must have a negative control and you must use a positive control so that you do not have any false positives or false negatives. So in the ELISA, which are the plate formats, not the strip formats, ELISA can be divided into direct ELISA and indirect ELISA. In direct ELISA, what we do is the antigen or the analyte is bound to the solid phase, and then you add an enzyme labeled antibody, wash off all the unbound, and then supply the substrate. And if you see a reaction, then the antigen is present and you can quantitate it. In indirect ELISA, we use 2 antibodies, one primary antibody and a secondary conjugated antibody, which is bound to an enzyme. And the enzyme will react with the substrate to produce a color which can easily be visualized. So in direct ELISA, you see you have an antibody, and the antibody is tagged with a substrate, an enzyme, and when the antibody binds, when the analyte binds to the antibody, this substrate, if you add the enzyme, you get a color and it says it is positive, and exactly what you saw in the lateral force tip. So you also have what is known as competitive ELISA, where the labeled antigen competes for the primary antibody binding sites with the sample antigen. And this is used in radioimmunoassay. Towards the end of this webinar, the competitive ELISA will be more, the explanation will be far better there with the radioimmunoassay. So then we have in the ELISA where we have what is known as sandwich ELISA. In sandwich ELISA, we generally use the coated plates, which are the 96 well plates, or they could be 8 well strips. Here, the 96 well plates or the strips are coated, already pre-coated with the antibody for the analyte in question. So now, if you make a solution or you take your sample containing the analyte and add it to this plate, because of the interaction, the analyte will be captured by the by the antibodies which have been coated onto this plate. Then you add a secondary antibody to it, which is linked with an enzyme such as horseradish peroxidase. And when you add the substrate, wherever the antibody has bound, it will react with the substrate. The enzyme will react with the substrate and a color will be formed. So this explains the sandwich ELISA. So here you have the sample. Now, the antibody which is coated in the plate is towards this target analyte. So you see the plates are coated with the antibody. We put the sample, and now only this analyte will bind. Then we do a wash step to wash away all the other analytes, and only the analyte which is specific for the antibody will be bound. Then you add a secondary antibody which is conjugated to an enzyme like horseradish peroxidase or the beta galactosidase, okay, reactions which, or with alkaline phosphatase, reactions which will produce a color. So now you add the secondary antibody. The secondary antibody now recognizes the antigen over here, and the binding will take place. Then another step of washing to remove the unbound secondary antibody, and then the substrate is added and the reaction allowed to proceed, and the color red. So the enzyme labels that we use in for the ELISA, they should have high specificity, they should have very high reactivity, they should be able to couple to the antigen antibody complex. So three enzymes that are commonly used are the horseradish peroxidase, alkaline phosphatase, and glucose oxidase. These are the enzymes which are generally conjugated with the secondary antibodies which are used in enzyme-linked immunosorbent assay. So in sandwich ELISA, the first step is to prepare a surface to which a known quantity of capture antibody is bound. That is, now you get ready-made plates with the bound antibody. You block any non-specific binding sites on the surface, then apply the sample which contains the analyte of interest, allow it to bind. Generally, we keep it overnight. You wash the plate so that you remove the unbound antigen, as you saw, and then you add the primary antibody as detection antibodies, which will specifically bind to the antigen. You wash the plate so that the unbound antibody enzyme conjugates are removed, and then you add a chemical which is used, or a substrate which is used by the enzyme to convert it into a colored product. And then you measure the absorbency of the plate wells. We have got what are known as ELISA readers, which are actually UV visible spectrophotometers, which can read the absorbance of each of these wells. So in indirect ELISA, as you see, so here is a 96 well plate, a part of it. The difference here is in the sandwich ELISA ending, the antibodies are already pre-coated in the plates. In the indirect ELISA, we do not use pre-coated plates. However, we take the sample and we allow the sample, the antigen, to bind. So the first step is to add the sample and to allow the sample to bind. So we add different quantities of the sample in each well, and we allow it to bind overnight. Then you wash out the unbound samples. You do not wash out. You actually see that the binding is complete. Then you add the antibody to the antigen, allow the antibody to bind to the analyte. You wash out the unbound antibodies. Now you have an antibody which is bound. You add the secondary antibody which has got an enzyme tag attached to it, and then you wash out all the unbound antibodies. So now you have only the enzyme tag bound to the antibody. You add the substrate reacts with the enzyme which is bound here, and what you get is a color. So this is the color that you see in the plates, and these colors can be directly read in the ELISA plate reader. So what you see in the wells over here is there is very little protein, and here you see that where you see the immense color, there is more protein. So generally in one well, we will add the standard in different quantities so that we can make a calibration curve, and from the calibration curve, you can find out the amount of the antigens. So the results are plotted where we use the concentration of the analyte on the x-axis and the absorption depending on the product of the enzyme reaction on the y-axis, and then the unknown value. With the absorption of the unknown value, you can calculate the concentration of the analyte of interest. Now you get the kits which give you all the instructions, including how much of antibody to be added, how many times to wash, etc. The ELISA readers also come with plate washers, ELISA plate washers, and they are automatically washed and emptied. And so there is no need of any manual interference for the ELISA technique, other than adding the sample in the beginning. So the general ELISA protocol, as I told you, is here. It includes the plate preparation, whether you're using indirect ELISA or thing, but if you're using sandwich ELISA, you already have the plate, so only you have to bind the antigen. Then you have the assay procedure for the enzyme, and finally it is the calculation of the results. The radioimmunoassay is a form of ELISA, of competitive ELISA, but instead of using an enzyme over here, we use a radioactive component, and generally we use tritium, that is H3, or we use iodine 125 or iodine 113 or carbon 14. So this is a very sensitive method. It is much more sensitive than the substrate-based or the enzyme-based enzyme assays. It is used to measure drugs, doping agents, especially in the food supplements, antigens, hormones, and etc. It can be used wherever an antibody can be produced. Okay, and it is used in drug detection, blood bank screening, and also for certain viruses and cancers. So the radioimmunoassay produces signals in response to the binding, and here radioisotopes are used. The most commonly used radioisotope is iodine 125 and iodine 131, easier for it to be introduced in the tyrosine residues. So this is the protocol for the radioimmunoassay. So here what you do is you have to first have a labeled analyte. So you have to prepare a labeled antigen. So you use either tritium or iodine to label the antigen. Now you take the labeled antigen, that is which has the radioactive label in it, and you add the antibody. Now the antigen recognizes the antibody, so you have the radio label binds to the antibody. That is the known amount. You add a known amount of the labeled radio labeled antigen to the specific antibody it binds. Now, to this, you add the unlabeled antigen or the sample. So now there is a competition between the unlabeled antigen and the labeled antigen for the sites on the antibody. So in this reaction, the unlabeled antigen will now compete and it will bind to the antibody, knocking off or displacing the label, that is the radioactive label. Then you precipitate the antigen antibody reaction, which is there, and you measure the radioactivity in the supernatant and the radioactivity in the pellet. And this tells you the radioactivity in the supernatant is the unbound or free antigen, and the radioactivity in the pellet is the bound antigen. So in the next step, it is you draw a graph of the bound versus unbound. So you have the unlabeled antigen, and you plot it against the counts of the bound versus the counts of the free. So what you get is a hyperbola. You will not get a straight line. So from the ratio of the bound to free, the unknown ratio, you can calculate the concentration of the unknown. So radioimmunoassay is a form of competitive ELISA, where the radio-labeled antigen and the unlabeled antigen compete for the antibody, and from the ratios of the bound to unbound, you can calculate the amount of analyte. So after determining the ratio of bound to free antigen in each unknown, the antigen concentrations can be read directly from this standard curve. So the next webinar is quality assurance and quality control on Monday.